Hello, everybody, and good afternoon. Thank you for joining me in this session. I want to start by first thanking the Ülker Center, the Ülker Foundation, and indeed the family for bringing us together in this virtual format. Of course, I'd love to be in Istanbul right now, and I hope I will get the opportunity to come back and visit sometime. I also would like to thank Gökhan Hotamishligil for his leadership in the center, our school here at Harvard, and indeed science in the United States and Turkey. It's great to be part of this here in our department. I also want to start by acknowledging that all the work that I'm going to share with you today is the product of a scientific partnership between myself and Bob Faris, shown here. And if you're interested about the lessons we've learned along the way, we've recently tried to put them down in an essay published in the JCI. And today I'm going to talk about work, particularly generated by three members of our laboratory, Jeon Chung, Maria Jesus Olarte, and Shuwei Sui, which of course built on the work of a number of other people coming through the laboratory. Now, the fundamental question we're interested in is really how cells and organisms deal with fluctuations in energy. And if you think about any living system is driven by flux of energy through its cells. There is constant energy intake and often a need to expand energy. And because both the sources and the need to expend energy varies greatly over time, systems and organisms have evolved the potential to temporarily store metabolic energy. And by and large, as you can see on this slide, they do this in the form of lipids, and much less so in the form of carbohydrates. This here is drawn roughly to scale for a healthy human to show how much of the metabolic energy in the body is stored as lipids as opposed to carbohydrates. And as I mentioned, this is not a property unique to humans, but indeed lipid stores can be found throughout life, including in bacteria and flies and yeast and mammalian cells. And of course, you all know that in humans, fat or lipids is mostly stored in adipose tissue, but it is also found in many, many other tissues, at least temporarily, for instance, after a meal in the intestine or in the liver or in the heart, for instance. Now, biochemically, the reason probably that fat has evolved as some major storage form is because the molecules in it, triacylglycerol, the neutral lipids, are very, very hydrophobic. And as a consequence, they exclude water. As you can see, of course, when you mix oil with water, what happens is that it forms a tightly packed phase. And this exclusion of water allows cells and organisms to very compactly store the metabolic energy. And on the cellular level, this occurs in highly unusual organelles that we now know as lipid droplets. You can see in this micrograph here that a lipid droplet, and schematically, it consists of a core of neutral lipids, particularly triacylglycerols, so where one glycerol backbone is esterified to three acyl chains, and depending on cell types, other neutral lipids such as sterol esters. And this organelle, this core is bounded by a monolayer of phospholipids into which specific proteins are embedded. You can also see on this micrograph that lipid triplets crosstalk with many other organelles in the cell, such as the mitochondria or the endoplasmic reticulum. Now, if you don't take anything away from my talk, it is that I want to at least leave you with the impression that the presence of lipid droplets essentially convert cells to emulsions in which the cytosol forms the continuous phase and the lipid droplets form a organic oil phase that is separated from the aqueous phase by emulsifiers and surfactants. And so this generates what we believe is really a fundamental problem for cells to deal with. Because if you look at this sort of experiment done in my kitchen this morning, you can see really the fundamental problem becomes that the oil is the most compact form to store metabolic energy. And but in a cell modeled here by this cocktail glass, really the water is where all the life happens. So the fundamental question then becomes, how does the life that happens in the aqueous solution deal with the oil? How does it make it the oil? And how does it ensure that it can utilize it later on? So the two topics that I want to cover today in my talk is like how cells generate the oil phase and how do proteins actually then get onto this newly formed organelle in order to help mediate the metabolism of the oil and other things. So concerning the first step, we've known for many years, starting from the work, for instance, pioneering work of Eugene Kennedy here at Harvard, that the enzymes, that there's an enzymatic pathway that generates triacylglycerol. This pathway starts with the activation of fatty acids, and then the sequential esterification of those activated fatty acids onto glycerol 3-phosphate, yielding eventually diacylglycerol, that is a polar lipid, and then finally triacylglycerol, which is a highly hydrophobic molecule. Particularly this last step really is responsible for generating oils, and it is catalyzed by two distinct enzymes that were initially discovered by Bob, and they are called DGAT1 and DGAT2. Those enzymes share a similar name because they catalyze diacylglycerol acetyl transferase activity, but they're otherwise unrelated in evolution. And you can see here that if you take cells and generate adipocytes in vitro, they form abundant oil stores here labeled with a red dye. But if you do not have either of the DGATs, you do not make any of the oil, even though the cells are present here. It's worth mentioning that those two enzymes are intriguing in their cell biology because they have different properties. For instance, DGAT1 localizes exclusively to the endoplasmic reticulum, here shown in red, and not so much anywhere close to lipid droplets in green, whereas DGAT2 appears to localize in many systems around lipid droplets. Now, how do these enzymes actually work? Turns out DGAT1 is conserved throughout evolution and forms a higher oligomeric structure, usually dimers or tetramers, but we have very little information of how these enzymes actually take the aqueous substrates and generate the oil phase for forming lipid droplet. So Shui Wusui in the lab used the emerging technology of cryo-electron microscopy to solve the structure of DGAT1, and he found this beautiful butterfly-shaped molecule that sits in the plane of the membrane. And here's one monomer, and you can see in the butterfly structure there were two molecules that came together. And in the middle of this, in the plane of the membrane, there is a catalytic residue buried, a histidine 415. And without going through much detail, the structure really revealed this beautiful system that takes from the cytosol molecules such as the glycerol 3-phosphate and the acyl-CoA, and most likely catalyzes the formation of the triacylglycerol right within the plane of the membrane. And then the structure revealed a lateral gate onto the plane of the membrane, and it is one possibility that this exit tunnel is used to directly eject the oil into the plane of the membrane. So in this model here, we would have the substrate enter from the cytoplasmic side, then catalysis occurs, and the product is released through this pathway. We do not have yet any information how DGAT2 operates, but of course are very interested in that. Now, if that's the first step here of the formation of an oil phase in cells, we start with forming these blisters. And we know that just generating oil in a membrane would be sufficient to eventually make a lipid droplet. But it is our belief, based on experiments over the last decade or so, that cells don't leave this up to chance, but instead have evolved machinery that really organizes this process in a particular place. And one of the key molecules for this process is a protein called BSCL2 or sapin, which was originally identified because it is mutated in patients with congenital generalized lipodystrophy. It was also identified roughly a few years later, actually, as a major factor in a number of genetic screens, for instance, from Rob Young and Joel Goodman's lab in yeast. And it's been shown that it forms large oligomeric complexes that localize to ER and lipid droplet junctions, and that in model system it is required for normal lipid droplets. Now here's an image from an actual patient fibroblast. This is a control proband, and you can see sort of in white here normal lipid droplets. But when you take cells from a person who suffers from this lipodystrophy, you can see that instead now the lipid droplets look far less organized, and instead of being forming these regular structures, there's all this schmutz here sort of in the side. This can be modeled in a variety of systems, and you can see, for instance, here a time course of lipid droplet formation in a wild type cell. This is a Drosophila cell. You can see at the beginning of the movie, this normal cell has a couple of pre-existing droplets, but then we form new ones here, and it appears to happen in a highly organized process. Yet when sapin is gone, there's a couple of pre-existing droplets, but now instead of this organized process, you start seeing all this little snowstorm here in the back of these tiny, tiny droplets that form in a very disorganized fashion. These sort of unorganized lipid droplets are sort of very small lipid droplets that can be found all over the cell that appear to be distinct from the endoplasmic reticulum and are just forming what we believe is ectopically, as I will show you. So sapin really got interesting to us when we start localizing the protein using genome engineering technology, localizing the endogenous protein. We found that localized to the endoplasmic reticulum, which appear to zip around the ER almost like if they were scanning the membrane. And this is true in Drosophila cells, and it is also true in mammalian cells. So what are these foci? Again, Shui Wu turned to cryo-electron microscopy and solved the structure of the sapin molecule, and he found this beautiful ring-like structure. So if you look at the topology of this protein, it sits in the endoplasmic reticulum membrane with two transmembrane domains in the luminal domain. In our initial structure, what's resolved is this luminal domain sitting or predicted to sit right underneath the membrane. So and the structure has two particular features. It has a hydrophobic helix in the middle and this beta sandwich fold. And in fact, each one of those luminal domains oligomerizes to give you that large dodecameric ring. And this is how we pictured it sits in the membrane, with the hydrophobic helixes positioned against or into the membrane, and the transmembrane domains, which were not part of our structure, to anchor it to the membrane. Now, this, of course, immediately suggested to us that maybe the way this protein functions to organize droplet formation is by sort of interacting with triacylglycerol through these hydrophobic helices during the lipid droplet formation. But when Jian Chung joined our lab, she realized something quite peculiar, and that is that this helix here is not only hydrophobic, but it is also conserved in sequence through evolution. And if it was just needed to be hydrophobic, this would be somewhat unusual. So Jian hypothesized that another protein factor might interact with this sequence, and indeed designed an experiment that would test this hypothesis by, in a number of experiments here. I just show you one. She did a pull-down analysis where we have either this hydrophobic helix or we do not have it. Pull down and ask, are there any specifically enriched proteins? And she found exactly one that was only present when this little helix was there. And she did a lot of control experiments that I don't have time to show you, but convinced herself and us that TMEM159 is a protein that specifically interacts with sapin. So what is that? It is a small membrane protein that has 4 putative trans or membrane embedded helices that was originally identified as a PPAR gamma target induced in the development of murine fatty liver as in a model. It also had a very high correlation in a genome-wide screen with phenotypes of sapin and maybe a homologue of some yeast proteins that were also postulated to be involved in lipid droplet formation. So Jian also purified the protein in a complex together with sapin. This is here the final step of this purification from a size exclusion chromatography. And you can see first that these proteins migrate together in a size exclusion chromatography and appear to form a stoichiometric complex. And what was really exciting to us is, like, when we purified sapin alone, we found no associated triglyceride, which was a little bit of a dent in our model that it would be involved in the initial steps of droplet formation. But as we started purifying the complex between the two proteins, we could see on a TLC that there was triglyceride associated with this complex. And this could be greatly increased if at the same time we induce droplet formation by including fatty acids into the media. And we know that this is triglyceride because it migrates at the same height of triglyceride, and its formation can be blocked by DGAT inhibitors. So this then suggested this really beautiful model that this complex together is involved in initial droplet formation. And this is further supported by our initial electron microscopy attempts, where we see that sapin alone forms a flat micelle. But if you have the complex, actually the luminal structure stays the same, but you now see sort of this bulge out in the membrane. And remember, this is where there is some triglyceride included. So we believe that this is sort of a trapped intermediate, in which LDAF and triglyceride together are sort of in the core of this, sort of giving us sort of the first view of a forming lipid droplet in its lens stage. So consistent with the idea that these two proteins function together, you can see on this slide here that both actually form complexes in the ER and mammalian cells, and those complexes co-localize to a large degree. And only at sites where you have both proteins present, these here are triple engineered cells to express three different tags, one on sapin, one on TMEM159, which we now renamed lipid droplet assembly factor 1, and one on the marker perilipin 3. And only when you have both of those proteins present can you form a lipid droplet. Sites that only have one or the other protein do not do that. And here you can see this process. We start out with a complex between the two proteins. And something fascinating happens that we don't fully understand yet is, as lipid droplets form, the sapin stays at the apparent neck with the ER, whereas the LDAF protein slowly migrates onto the surface of the lipid droplet, yielding a structure in which you have the ER background, the sapin at the nexus, and LDAF on its surface. And consistent with an important function of LDAF, where we knock the protein out, we again have disorganized and less efficient lipid droplet formation. So this is leading to our current working model for this process, in which triglyceride synthesis provides molecules that are then phase separating in this amazing machinery of sapin and LDAF. Just as a side note, sapin, there's a dodecamer 12 molecules that gives you 24 transmembrane helices. LDAF has 4 per molecule potentially, and seems to be stoichiometric. So you have upwards of 60 transmembrane domains in this complex here. And this then basically allows phase separation, the initial lipid droplet formation from the ER. At least in my mind, this is somewhat reminiscent of the model for phase separation during protein folding pioneered by work from Ulrich Hartl and Art Gorwich, in which the chaperoning complex really generates an environment in which the molecules, in this case of a protein chain, are allowed to preferentially interact with itself, and thereby generating a folding conducive environment. And we think that maybe sapin might have a similar function in this complex to allow triglyceride molecules to preferentially interact with themselves, and thereby this complex promotes phase separation and the formation of the lipid droplets. If you don't have that, you cannot generate these initially well-organized lipid droplets, but because you still make oil and it's still water in a cell, eventually you form these ectopic lipid droplets. I do understand that this is, of course, only a model, and we are hard at work to further test this and its alternatives as we go on. I want to shift gears here for a second and bring sort of the second fundamental question back into the light. And it is, once you made that lipid droplet, of course, you need to mobilize the fat, you need to put things into the bank, you need to withdraw things, and all of this is happening through the action of proteins that localize specifically to lipid droplets. Through the work of many laboratories, including ours, we now know that there is upwards of 100 proteins in mammalian cells that are specific to lipid droplets. Some of them are involved in processes that you might imagine should happen on the surface of lipid droplets, such as triacylglycerol and fat synthesis, or lipolysis, the process of breaking down lipids from the lipid droplet. There's a number of others that make perfect sense, but we continue to discover proteins for which we do not understand why they localize to lipid droplets in many systems, and whether and how the lipid droplet localization may function. So there's a lot of room for discovery here, but suffice it to say this really makes it very important to understand how the proteins actually specifically get to the fat and how they deal with that interface there. And the work on this problem has revealed that essentially there are two pathways. One targets proteins from the cytosol to the lipid droplet surface, and we call this pathway cytol for cytoplasm to lipid droplet targeting. And the second pathway in which proteins are initially inserted into the endoplasmic reticulum, and then from there migrate to the surface of lipid droplets. Now, this second pathway in particular relocalizes or has a whole number of clients that are involved in triglyceride metabolism, including, for instance, GPAT4, the first and rate limiting step, or catalyzing the first and rate limiting step for glycerolipid synthesis, and ATGL, the major lipase that takes the things apart again. It also has a number of very important proteins that are poorly understood yet, but medically relevant, such as HSD17B13. Now, all of these proteins appear to start their life cycle in the endoplasmic reticulum. You can see this here in a cell where we labeled GPAT4 in red and the ER in green, and you can see that both proteins localize very nicely to the ER as in cells that do not have lipid droplets. But as soon as we induce the formation of lipid droplets, the protein GPAT4 relocalizes to the surface of lipid droplets. And as these lipid droplets grow, you can see it basically covering this in a ring-like fashion. We believe this really is the surface of the lipid droplet because we used antibodies against the endogenous protein to see that by immunogold labeling, this protein is localized to the surface of droplets, but not the endoplasmic reticulum that is close by. So we really try to understand GPAT4 as a model for this targeting pathway. And particularly, we narrowed it down to sort of two particular substrates that we found useful. One is the full-length protein. This is the enzyme that synthesizes lysophosphatidic acid. It has two cytoplasmic domains, and in between, what we believe is a hairpin that anchors it in the ER. And we can sort of map a minimal domain that's just this anchor, 54 amino acids that we labeled called life drop, that is sufficient to get you onto lipid droplets. And one property of those two substrates that we use as models here is that really puzzled us for a few years is that they both start out in the ER. You can see this labeled in red. But if you start lipid droplet formation, they behave somewhat differently. So the full-length protein stays in the ER, whereas life drop goes onto droplets as soon as they are formed. Yet at the end of this reaction, when there is abundant lipid droplets, both proteins actually end up on the surface. So of course, this immediately questions us like, okay, if the protein's in the ER and the droplet forms in the ER, why does the full-length protein not immediately go onto the lipid droplet? And we now believe that an answer comes from the complex that I just showed you, the SAPIN-LF complex, and particularly SAPIN, that's at the lipid droplet formation site, and that these many transmembrane domains that are as part of this here effectively provide a barrier to access the lipid droplet. And consistent with this, if you take out SAPIN, you can see that the wild-type protein here normally localizes to the ER in a wild-type cell, but if you take out SAPIN, actually this protein can go onto the lipid droplet during their formation. In both cases, the proteins end up on lipid droplets at the end of the time course. So we think that SAPIN really provides sort of a barrier that at least prevents some, maybe not all proteins to access the lipid droplets during their formation. So if that's true, then how do the proteins later accumulate on lipid droplet? And here's another, what I consider really important finding made by an observation made originally by Florian Wilfling when he was in the lab. And that was that if you follow the localization of this protein over time, many hours after the initial droplet has formed, nothing seems to happen. But then within a very short period, almost if there was a trigger, the protein can localize to one or a few lipid droplets in a somewhat asynchronous manner. And so then, of course, the question becomes immediately, how does the protein do that? And what we found in FRUP experiments is that during that time point, there appear to be abundant connections between the endoplasmic reticulum and the lipid droplet. In these experiments, what we bleach the signal on the lipid droplet, and you can see that it recovers apparently through these connections between the ER and the lipid droplet. You can visualize these connections by cryotomography. You can see the ER studded with ribosomes that's in direct continuity with the endoplasmic reticulum, with the lipid droplet. And we think normally that every droplet may have one sapin connection, but you see many, many more. So these appear to be independent of sapin. And this is just a rendition. So we think this provides the pathway for how proteins can travel from the ER to the lipid droplet. But why, if there is a road, do the proteins actually travel and accumulate on droplets? And this is the problem that Maria Jesus Olarte tackled. And first, what she found is that this is a property that is encoded apparently in the sequence of this hairpin in GPAT4. Because if you take the wild type sequence, it is sufficient to bring a protein to lipid droplet. But if you scramble the sequence, the protein still inserts in the ER, but now it does no longer accumulate on the droplet surface. And I don't have time to show you all the data that she generated, but I want to leave you with the model how we currently believe this occurs, which is that the proteins are inserted into the endoplasmic reticulum under a strain in which positively charged residues anchor the hairpin to both sides of the membrane. And this positions large hydrophobic residues into an energetically unfavorable position in the middle of the bilayer. As the proteins transition to the surface of lipid droplets, this restraint here is released, leading to the accumulation of tryptophans on the surface of the monolayer, leading to an energetically more favorable state on the membrane. And just as for other experiments I've shown you today, I'm only going to refer to GPAT4 here, but we've got consistent and very similar results for other proteins. So we think from this is that there's actually two Ertl pathways. There's one early pathway that occurs, for instance, for LDAF and allows targeting as droplets form. And then probably the more prevalent one is the late Ertl pathway, which occurs through these bridges. And of course, we're working on to really understand how these bridges are established and how they allow trafficking when you have those two different pathways. I also want to leave you with a sense of wonder, and really that I think we're scratching the iceberg on a fundamental question of biology here. We do not understand, for instance, how sapin works for some proteins but not others, to exclude them during their formation. Which protein signals mediate the axis or prevent it? How is this late Ertl pathway triggered? I showed you an experiment in which you get like rapid accumulation on some droplets, but we don't understand how, when, and why this occurs. How persistent are these bridges and how are they actually made? Are they constitute one-way streets, or can you also get proteins off the lipid droplet? And this is just to say that despite the significant progress I think we've made as a group and as a field, there's many, many exciting questions left. Now, I acknowledged most of the people as I was talking. I want to also highlight Henning's contribution to this work that I showed today. We have a long-standing collaboration with Shinra Liu and Morgan Graham at Yale, and for a lot of the molecular dynamics simulation that I've showed today, we collaborated with a group of Greg Wood, Jessica Swanson, and Si-Young Kim at Chicago and Utah. And of course, I want to acknowledge our funding sources, and in particular for this, the National Institutes of Health, as well as the Howard Hughes Medical Institute. And with that, I'd be happy to take any questions you may have. Thank you very much.